A rat infected with Toxoplasma gondii doesn't just lose its fear of cats. It becomes attracted to them, not indifferent, not less cautious. Actively drawn toward the scent of cat urine the way a healthy rat is drawn toward food or a mate. That's not a metaphor. That's the result of a parasite rewriting the rat's brain chemistry to serve its own reproductive agenda. And it's been doing this for millions of years. Toxoplasma gondii is a single-celled parasite, a protozoan, smaller than anything you could see without a microscope. It needs two things to complete its life cycle: an intermediate host, any warm-blooded animal, and a definitive host, a cat. The reason it needs a cat specifically is mechanical. Toxoplasma can only sexually reproduce inside the intestinal lining of a felid. Any felid: domestic cat, leopard, jaguar, cheetah. Outside that environment, it can replicate, but it can't complete its cycle. It can't produce the oocysts, the hardy egg-like structures it needs to spread. So it does something that has no precedent in classical parasite biology. It engineers the encounter. The standard model of parasitism is passive. A tapeworm doesn't care what you do. It sits in your gut and absorbs nutrients. A malaria parasite needs a mosquito to move between hosts, but it doesn't alter the mosquito's behavior to make that more likely. Toxoplasma does something categorically different. It reaches into the central nervous system of its intermediate host and adjusts the animal's behavior to increase the probability that it gets eaten by a cat. The parasite doesn't want the rat to survive. It wants the rat to fail in a specific, directed way. Here's what that looks like in practice. A healthy rat avoids the scent of cat urine. This is not learned behavior. It's innate, hardwired. A rat raised in a laboratory with zero exposure to cats will still freeze, retreat, and show elevated stress hormones when exposed to cat urine. The aversion is baked into the architecture. In 2000, researchers at the University of Oxford exposed infected rats to cat urine and measured their response. The infected rats didn't just show reduced fear. Some of them showed attraction. They spent more time near the scent source. They investigated it. The behavior that should have kept them alive was running in reverse. Manuel Berdoy led that study. What he published wasn't a curiosity. It was a mechanism problem. How does a single-celled organism with no nervous system alter the behavior of one that has one? The answer took another decade to fully emerge, and it's stranger than anyone anticipated. Toxoplasma forms cysts. When it enters an intermediate host, it travels through the bloodstream and parks itself in long-term tissue: muscle, liver, and most critically, the brain. Inside the brain, it preferentially targets the amygdala. The amygdala processes fear. It's the region that fires when the rat detects a threat and generates the biochemical cascade that produces avoidance behavior. Toxoplasma doesn't destroy it. It infiltrates it and alters what it does. Inside the cysts, the parasite carries genes for an enzyme called tyrosine hydroxylase. That enzyme is a precursor in the dopamine synthesis pathway. Dopamine is the neurotransmitter most associated with reward, motivation, and approach behavior. The parasite is producing the molecular raw material for reward signaling inside the fear processing center of the brain. The rat's amygdala starts associating cat odor with something closer to reward than threat. The fear circuit gets rerouted. Ajay V.S. and Robert Sapolsky at Stanford confirmed this in 2007. They mapped the neural activity of infected rats exposed to cat urine and found that the regions associated with sexual arousal were activating in response to the scent, not the fear regions, the regions the rat's brain uses to process attraction. The parasite had taken the neural pathway for sexual motivation and aimed it at the thing most likely to get the rat killed. From the outside, the infected rat doesn't look sick. It doesn't move differently. It doesn't display lesions or obvious distress. It goes about its life normally until a cat is nearby, and then its behavior shifts in exactly the direction the parasite needs. This is where the biology stops being only about rats. Toxoplasma gondii infects approximately 2 billion humans. Roughly a quarter of the global population carries it. In some countries, infection rates exceed 60%. The vast majority of those people have no idea. In a healthy immune system, the initial infection produces flu-like symptoms that resolve in weeks. The cysts form, the immune system walls them off, and life continues. No obvious illness, no treatment required. Classified as a chronic latent infection, meaning the parasite is present, contained, and presumed inactive. That presumption is where it gets complicated. Jaroslav Fleger, a Czech evolutionary biologist, became interested in toxoplasma in the early 1990s after noticing subtle shifts in his own risk perception. He was infected. He began running large-scale studies on infected versus uninfected human populations, measuring personality traits, reaction times, and behavior patterns. The results were contested and are still debated, but they were not nothing. Infected men in his studies showed slightly reduced reaction times and modestly elevated novelty-seeking behavior. Infected women showed different patterns. The effects were not large. They were not uniform, but they were statistically present in populations large enough to take seriously. The mechanism in humans would be the same as in rats. Toxoplasma forms cysts in the human brain, preferentially in the amygdala and prefrontal cortex. It carries the same tyrosine hydroxylase gene. It produces the same dopamine precursors. Whether the behavioral effects are large enough to matter in a human population, or whether they're simply statistical noise from a weakly powered study, is genuinely unresolved. What is resolved is the mechanism. The parasite is doing the same thing in human neural tissue that it does in rat neural tissue. Whether the outcome is equivalent is a different question. There's a further complication. Schizophrenia researchers noticed something in the data decades ago. Individuals diagnosed with schizophrenia have statistically elevated rates of toxoplasma antibodies compared to the general population. The association has now been replicated across multiple countries and study designs. E. Fuller Torrey and Robert Yokin published a meta-analysis in 2003 synthesizing the findings. The association doesn't prove causation. Schizophrenia has multiple causes and most infected people never develop it. But the elevated antibody rates appear consistently enough that some researchers have proposed toxoplasma as a contributing factor in a subset of cases, potentially through its dopamine pathway effects in vulnerable individuals. The parasite also interacts directly with immune function. During pregnancy, a new infection can cross the placental barrier and cause serious damage to the developing fetal brain. This is why pregnant women are advised to avoid handling cat litter. A chronic latent infection acquired before pregnancy appears to carry much lower risk. The danger is new infection during a period when immune suppression is already elevated. Toxoplasma exploits the window. The organism itself has been on Earth for an estimated 1 to 3 million years. It predates modern cats by some margin, having co-evolved with ancestral felids. The manipulation behavior it runs in rodents is not a recent adaptation. It's a refined, stable strategy that has been running long enough to become precise. The rat's fear circuitry didn't evolve to be hijacked this way. Toxoplasma evolved to hijack it. The parasite has been running this experiment on mammalian brains across geological time, and the mammalian brain has not caught up. What makes this particular organism difficult to categorize is the specificity of the target. Most parasites that alter host behavior do so bluntly. They produce general lethargy, or generalized risk-taking, or reduced immune signaling. Toxoplasma is surgical. It doesn't make the rat reckless across the board. The infected rat still avoids open spaces, still responds normally to novel objects, still shows normal social behavior. The only thing that changes is its response to cat odor specifically. The manipulation is narrow, targeted, and serves one purpose: getting the rat into a cat's mouth, a parasite smaller than a human red blood cell, with no nervous system, no muscles, no brain, has developed a strategy that requires understanding, at some functional level, the relationship between a rat and a predator, and intervening in that relationship in exactly the right way at exactly the right point. It doesn't know it's doing this. It has no awareness. The behavior emerged through selection pressure over millions of years of trial and error on a scale too large to visualize, but the result is a system that looks from the outside almost like planning. The rat that walks toward the cat isn't broken. It's been reprogrammed, and the thing that reprogrammed it has been doing this longer than our species has existed. Thanks for watching. See you on the next one.